Hello and welcome. My name is Henrique Valim, and in this talk, I hope to bring you some insights into circadian clock systems, as well as to highlight the circadian clock as a lens through which to explore environmental adaptation in lichens. Although all organisms are capable of responding to diurnal environmental stimuli, most also feature an endogenous timekeeping mechanism known as the circadian clock that allows them to maintain rhythmicity in the absence of these outside stimuli. The circadian clock allows organisms to respond to a variety of stimuli in a time-contextualized manner. For example, organisms can limit their sensitivity to certain stimuli within a time window, a process known as gating, in order to avoid responding to some stimuli at time points when these stimuli do not affect fitness. Evidence for the functional importance of the circadian clock comes to us from a variety of angles. In the first place, circadian clock components are remarkably well conserved throughout the tree of life, and the circadian clock appears to have evolved independently in at least 3 separate lineages, in animals and fungi, in plants, and in some prokaryotic lineages like cyanobacteria. Functioning circadian rhythms also confer fitness advantages, and disrupting these rhythms or the molecular components of the circadian clock machinery can lead to deleterious outcomes. Finally, functional circadian systems can be lost in arrhythmic contexts in which the clock does not confer any fitness benefits, as has been observed for populations of cavefish as well as for Arctic mammals such as reindeer. A small but growing body of research explores the potential role of interacting circadian clocks in holobiont and symbiotic systems. These studies have ranged from corals, in which the phenotypes of both the coral and its symbiotic dinoflagellates display circadian rhythmicity, to the gut microbiota of mammals, where microbiome composition and abundance both display circadian rhythmicity and can be disrupted by jet lag or manipulation of circadian clock components. However, although the circadian system of fungi, algae, and cyanobacteria individually have been studied in a variety of model organisms, virtually nothing is known about the circadian clocks of lichens. There are a variety of potential analogous roles, however, for the circadian clocks of both the photobionts and the mycobionts. In the case of photobionts, the circadian clock closely regulates photosynthetic activity. Given that lichenized fungi are known to associate with different algal strains across climatic gradients, algal circadian clock genes may harbor differentiated alleles across climatic zones. And in the case of the mycobiont, the circadian clock may play an important role in anticipating and dealing with harsh changes in abiotic conditions throughout the day, as well as from season to season. A further complicating element in the lichen circadian system is the microbiome. Microbiomes vary rhythmically for a variety of functions, and these may have different benefits to the other components of the symbiosis. Before exploring the holobiont circadian clock in all of its complexity, though, we must first understand who are the individual players involved within each symbiont circadian clock. Because of the fungus's important role in the lichen symbiosis, as well as the comparatively better elucidated circadian clock of fungi more generally, we first focused our efforts on identifying the components of the mycobiont circadian clock. The red bread mold, Neurospora crassa, has served as an important model system for circadian biology for many decades, and therefore has a well-elucidated circadian clock mechanism. At the core of this mechanism is the interaction between the genes white collar 1, white collar 2, and frequency. White collar 1 and 2 heterodimerize to form the white collar complex, which regulates the transcriptional activation of frequency gene. Frequency, in turn, indirectly targets the white collar complex for degradation, forming a rhythmic feedback loop common in many circadian systems. Both the white collar complex and frequency regulate a variety of other downstream targets in Neurospora crassa and in other filamentous fungi. And thus we ask whether we find these particular homologs for these 3 genes in the genomes of lichenized fungi. In particular, We looked for these three genes in the mycobiont of the lichen Umbilicaria pustulata. Umbilicaria pustulata is a rock-dwelling lichen with a wide distribution. Although the Trabuxia photobiont associated with Umbilicaria pustulata has been isolated, all attempts to culture the mycobiont in isolation have failed thus far. In order to sequence the mycobiont, therefore, we previously used a metagenomic approach combining both long-read and short-read sequencing to reconstruct the mycobiont genome. This hologenome sequencing has provided a very high read coverage of the mycobiont and a high-quality assembly in which to search for these circadian homologs. We identified the putative homologs of frequency, white collar 1 and white collar 2 by blasting the Neurospora crassa proteins against the Umbilicaria pustulata genome assembly. In order to contextualize the phylogeny for each gene, we included homologs of each circadian clock gene here from the basidiomycete shown in red. We further used the Umbilicaria pustulata protein sequences to search for homologs in other lichenized fungal genomes, identifying 3 other lichenomycetes, the Cladonia species, and 2 wolf lichens shown here in green. We also identified homologs in the more distantly related lichenized fungus of the genus Endogenous shown here in blue. We further included a variety of fungi from other lineages as well as Neurospora crassa. Interestingly, while we identified homologs of white collar 1 and 2 in each species, we could not identify a frequency homolog in 2 of the lichenized fungal species, the Cladonia and the Umbilicaria species. Although there is some evidence in the literature for lost homologs of frequency in other fungal lineages, it's unclear if this is an artifact of the genome assembly, given frequency's relatively high level of conservation across a variety of lineages, as well as the central role of frequency in the fungal circadian clock. A look at the alignment of each of these proteins does, however, reveal that white collar 1 and 2 are more well conserved than frequency. And these areas of high sequence similarity are concentrated on known functional domains of each of these proteins. It therefore seems feasible that the Umbilicaria pustulata homologs are functional circadian clock proteins. To further explore whether these genes may maintain some functional relevance for Umbilicaria pustulata, we investigated the variation in these 3 genes across 3 elevation gradients in Sardinia and Spain, in which pooled population samples of 100 individuals had been previously harvested from 15 sites across all 3 gradients. These 15 sites fell within 2 sets of climactic zones, either a cool temperate zone above roughly 800 to 900 meters above sea level, and a warmer Mediterranean zone below that. To look for strongly differentiated alleles in these 3 clock genes between these 2 climate zones, we looked at pairwise FST scores at all sites across the 3 gradients in the temperate climate zone and all sites across the 3 gradients in the Mediterranean zone. We compared both within each climate zone as well as between climate zones to identify patterns of differentiation. We would expect that if there are patterns of differentiation between the 2 climate zones, genetic variation should be much higher across the climate zones, which you see here in the red, rather than within each climate zone, which you see here in the blue. As expected, we did observe that genetic differentiation was much higher across climate zones than within each climate zone itself. Looking at these gene-wide genetic differentiation scores, we further asked whether this differentiation was localized in particular SNPs within each gene, such as in the functional domains I previously showed for each protein. In order to test then whether any differences in allele frequencies at particular SNPs are statistically significant, we calculated Fisher's exact test for each pairwise FST score. And here we see a visual aid at a cutoff of 0.05 to bring our attention to loci of interest. Given the gene-wide results, we would expect to see low differentiation within each climactic zone throughout each gene, while we may have expected to see islands of strong differentiation at particular SNPs associated with the fungal domains of each protein. As we expected, we saw low levels of genetic differentiation across all SNPs for each gene when looking at pairwise contrast within each climactic zone in the blue. And when looking across climactic zones, we observed SNPs with high levels of differentiation in both frequency and white collar 1, as well as 2 highly differentiated SNPs in white collar 2. Interestingly, however, many of these SNPs with strongly differentiated alleles were located outside of the functional domains for each protein. Upon closer inspection, we saw that many of these SNPs are located in intrinsically disordered regions found in all three proteins. These intrinsically disordered regions are known to be associated with protein stabilization and the formation of dynamic protein complexes. And they are known in particular to be involved in the stabilization of frequency protein during proper circadian function in Neurospora crassa. These results provide an interesting potential mechanism by which natural variation in circadian clock loci may yield circadian clocks adapted to different conditions across climactic gradients. Having now identified the putative components of the Umbilicaria pustulata core circadian clock, our next steps would be first to determine whether these genes function as expected in vivo, either through time series or through blue light activation experiments, as well as to explore the extent to which the pustulata genome is circadian regulated. Ultimately, we can investigate the patterns of variation and covariation in the photobionts of Umbilicaria pustulata in order to begin to understand how the circadian systems of fungi and algae vary along elevation gradients. We can then ask whether this pattern mirrors genome-wide patterns of distribution and how it contributes to the ability of the lichen symbiosis to expand the neutralistic niche along climactic gradients. And with that, I'd like to thank you for your attention, and I look forward to your questions.